योगः कर्मसु कौशलम्.
Hello everyone, hello my dear teachers and professors. Welcome to the discussion on nanotechnology. Today our discussion topic is role of nanotechnology in life science research. My dear friends, myself Himanshu Bariya and I am from Hemchandracharya, North Gujarat University, Patan. So today we are discussing about some important role of nanotechnology in life science research. We are today here about nanotechnology. It is a very emerging science today, and in recent years the nanomaterials have emerged as a novel type of material, as we know that. So this nanotechnology is the latest technology with options for utilization in different fields like biology, medicine, chemistry and physics. Here you can see the one figure. Due to having various shapes and structures such as nanorods, liposomes, dendrimers, nanoshell, quantum dots, etc., the nanomaterials appear as the most stable materials nowadays. These nanomaterials have a unique electrical and optical properties that can be synthesized by different ways at low cost and have wide applications in several interdisciplinary branches of science. As we know that life science is a general term which include all the subjects of the biological sciences including botany, zoology, biochemistry, biotechnology, microbiology and environment science. You can see in this figure there are various nanomaterials display over here, that water molecule, glucose molecules, antibody, viruses and bacteria. Now what we consider as a nanoscale. So the size from 1 to 100 nanometer, we consider it as a nanoscales. So here in the bottom there are synthesized nanomaterials are there, liposome are there, dendrimers are there, gold nanoshells, quantum dots and fullerene. These are the synthesized nanomaterial you can see in the figure. Now the term nanotechnology was first given by the Taniguchi in 1974. He was working with a very minute particle which have a size of 100 nanometers. As we know that nano means dwarf, is the Greek prefix which refers to the very small, which in terms of nanoparticles, and it can refer to a size up to 10 to power minus 9 meter. That means 1000 millionth of the meter. Okay, so nanoparticle belong to the nanoscience in which nano size molecules, which molecules, the molecules having a 1 to 100 nanometers are utilizing through practical application using the devices. Okay, as we know the Taniguchi gave a term nanotechnology in 1974. In recent year, according to the National Nanotechnology Initiative, that is NNI, United States, nanotechnology is defined as a field of science, engineering and technology where materials are practiced at the nanoscales, that means 1 to 100 nanometer, using a unique phenomena in a wide range of biology, physics, chemistry, medicine, etc. Okay, so the most important property of this nanoparticle are their size. As we know, as the size decreases, the increase in the reactions or surface area. So the most important properties of these nanoparticles are their size, which can manipulate the physicochemical and optical property of a particular substance. Nowadays, different nanoparticles such as gold, silver, nickel, titanium, etc. are synthesized in various shapes and colors for the delivery of chemical, biological sensing, biomining. It also include a gas sensing, capturing of carbon dioxides and other related applications. You can see in this figure, I show here the metallic nanoparticles like silver, copper, gold, iron, zinc, etc. So today these metallic nanoparticles play a very important role and perform a varying functions in different fields. Like silver and gold are mostly utilized because it is easy to synthesize, while copper, zinc, selenium, platinum are costly also, as well as it having some limitations. Okay, so now the nanoparticles are composed of three layers. As you can see in this figure, the outer layer is there, the blue layer is there in blue color, and center part is a core. So nanoparticles are composed of three layers. The first layer known as surface layer. Okay, you can see here the surface layer. The surface layer, which is composed of various type of small molecules, surfactants, metal ions and polymers, which functionalize the nanoparticles. Means depend on the attached molecules on the surface layer, the nanoparticle is get functionalized, right? Now the second layer consists of a shell layer. You can see here the NP cell. Okay, it is a shell layer composed of different chemical materials as compared to the core, and the core is the central part of the nanoparticle and generally refers to the nanoparticle itself. So due to such remarkable characteristics, these materials, which material? Nanomaterials, gain considerable interest from the researcher in multidisciplinary areas. In this discussion, I provide a common overview related to nanoparticle, such as their different types. You can see in this slide methods for synthesis, characterizations, properties and their biological applications. As we know, there are many type of nanoparticles are there. It is reported worldwide. There are certain methods for the synthesis of such nanoparticles. There are certain sophisticated methods through which we can characterize the synthesized nanoparticle, and these nanoparticles having a various biological applications. Okay, so here we will see one by one. So first we will discuss about a different types of nanoparticle. In these slides you can see a different type of nanoparticle, that is organic nanoparticles, hybrid nanoparticles and inorganic nanoparticles. So recently various categories of nanoparticles and their derivatives have been reported to have effective antimicrobial properties on the basis of their size, morphology and chemical properties. Now we are talking about the organic nanoparticles. You can see in this figure the top three, that is polymerical nanoparticle, liposome and dendrimers. These three are organic nanoparticles. Here I also take another images that include a dendrimer, liposome and micelles. These three are organic nanoparticle. Organic nanoparticles are solid particles ranging between 10 nanometer to 1 micrometer, that is 10 to 1000 nanometer in diameter, and consist of organic compounds like polymeric or lipids. So these three compounds like dendrimers, liposome and micelles are made up of polymeric or lipid. Organic nanoparticles receive little attention as compared to inorganic nanoparticles. Okay, so these organic nanoparticles or compounds are inherently and ultimately slow soluble in water. So they are slowly soluble in water or in aqueous environment, as compared to the inorganic counterparts. But here the organic nanoparticles will not remain in the environment for a long period, which makes them environmentally friendly. So what we have said, the organic nanoparticle is more environmental friendly. There are some well-known organic nanoparticles that have been reported, such as dendrimers, micelles, liposomes, and ferritin, that showed some characteristics properties such as non-toxicity and biodegradability. And micelles and liposomes have a hollow core. You can also tell it as a nanocapsule, and they are considered as more sensitive to thermal and electromagnetic radiations such as heat and light. Therefore, this unique characteristics makes them an ideal alternative for drug delivery. The drug carrying capacity, stability, and delivery system of organic nanoparticles, either in the form of entrapped drug or adsorbed drug system, determines their field application and their effectiveness. So these are all are about the organic nanoparticles. So ultimately these all organic nanoparticles are widely used in pharmaceutical industries. Okay, now moving towards the carbon nanomaterials. These carbon nanomaterials are very important. The carbon nanomaterials vary in shape, size, and functions, as we all know that. There are three categories of carbon nanomaterials. They are mostly carbon nanotubes, graphene oxides, and fullerene. Okay, here you can see in the figure the multi-wall carbon nanotubes are there. Single wall carbon nanotubes is there. There is a one nano horn. There is a bulky balls like structure, that is fullerenes. Carbon black nanoparticles is there, and nano graphite is there. It is also known as the graphene oxides. So the wall of carbon nanotubes can be single or multi, whereas graphene oxides are oxidized or reduced in form, and fullerenes are bulky balls like structure. Okay, so these type of carbon nanomaterials are used in textile engineering and medicines because they have showing a antimicrobial activity against certain bacteria, fungi, and also have demonstrated and investigated as a plant growth enhancer. So such type of carbon nanomaterials that you can see in this figure, like carbon nanotubes, graphene oxides, and fullerenes, opens a new area of microbiological research by uncovering microbial growth inhibition mechanisms. We know that certain bacteria and certain fungi are inhibited by this type of carbon nanomaterials. So certain forms of carbon nanomaterials can be produced for antimicrobial activity at relatively low cost, and it attracts researcher to develop an evaluation study for carbon nanomaterials in agriculture and other fields. So these type of carbon nanomaterials are widely used in such industries like pharmaceutical, medicines industries, and textile engineering. So this is about the carbon nanomaterials. Now move forward on the inorganic nanoparticles, as this figure is also discussed earlier in a certain slide. In one slide, the organic nanoparticles include all the metal nanoparticle. So metal based nanoparticle, metal oxide based nanoparticle are including in this inorganic nanoparticles. Nanoparticles composed of metal and metal oxides are generally classified as inorganic nanoparticles. Now we are discussing about the metal based nanoparticles. The nanoparticles have characteristics properties such as size as low as 10 to 100 nanometers, pore size, high surface area to volume ratio, surface charge and density, amorphous and crystalline structures, spherical or cylindrical in shape, a different color, high reactivity, and their sensitivity to environmental factors like moisture, air, heat, and sunlight. So, metal based nanoparticles are synthesized from metals such as aluminum, cadmium, cobalt, copper, gold, silver, and zinc, etc. And they can exist in solutions. You can prepare a solution of this nanoparticle, as well as you can prepare a powder of this nanoparticle by the lyophilization process. So such type of nanoparticles, which nanoparticle, metal based nanoparticle, have gained much attention in pharmaceutical industries for their use in manufacturing medicines. So you can see that the certain medicines are also having a nanomaterials. This nanoparticle can be modified by altering their chemical groups to binds with antibodies. So some noble metals such as silver minus Au, that is gold, and platinum, synthesized nanoparticles have specific property which were used in biomedical field to cure certain diseases. Therefore, these nanoparticles used to prepare drugs that have anti-cancer, radiotherapy enhancement, drug delivery, thermal ablation, antibacterial, diagnostic assay, antifungal, gene delivery, and many other properties. So this is about the inorganic nanoparticles, and these nanoparticles were found useful in like gold nanoparticle. Gold nanoparticles are found useful in identification of different microorganisms by detecting and evaluating DNA and identifying the protein interaction from the biological samples. So this is about the gold nanoparticle. Similarly, the silver nanoparticle combined with known antibiotic like amoxicillin, penicillin, vancomycin, and erythromycin shows antibacterial activity against the pathogenic strains. Moreover, the Ag nanoparticle, that is silver nanoparticle, play a very important role in the biomedicine, performing cell imaging, cancer therapy, genetic delivery, drug delivery, and different disease diagnosis. There are certain metal oxide based nanoparticles are also there. The metal oxide based nanoparticles such as Ag2O, O means oxide, FeO, MnO2, CuO, etc., enhance their activity and were found to have a potent antibacterial activity. The metal oxide based nanoparticles can be synthesized at the low cost using the simply accessible material and can also be utilized in food processing and environmental conservation with biomedical uses. This type of nanoparticle have excellent properties when compared with their metal counterparts. So these are about the inorganic nanoparticles. Now in this figure there are tremendous application of the nanoparticles like textile industries, biomedical sector, healthcare, food, agriculture, industrial, electronics as well, and environmentals. And each and every sector have their unique applications of the nanoparticles. Now how such type of nanoparticles are synthesized? So here we are discussing about the synthesis of nanoparticles. So there are several methods that can be employed for the synthesis of nanoparticles, which are most often divided into two main categories. You can see in this figure, that is bottom up method and top down methods. You can see the upper arrow is a bottom up approach and the downward arrow is a top down approach. Now what happened in both the methods? In the top down methods, we are discussing about the downward arrow. In the top down method, Nanoparticles are synthesized by decomposition of larger units into smaller units like grinding mills. Okay, and these smaller units are further converted into appropriate nanoparticle. Okay, so this method is followed by various types of processes such as mechanical milling, mechanical attriation, mechanical grinding, and some physical methods like thermal evaporation, thermal pyrolysis, and nanospraying. Okay, now while in bottom up synthesis, now the second one, upward arrow. While in bottom up synthesis, nanoparticles such as polymerisomes, liposome, vesicles, polymer conjugate, capsules, polymeric nanoparticles, and dendrimers. These type of nanomaterials or nanoparticles are synthesized by several processes like sol gel method, green synthesis, spinning, and chemical vapor depositions. It also includes certain chemicals and biological methods including polymerization, oxidation, reduction process, and in biological methods they are using plant extract, bacteria, fungi, and algae extracts. So among these all, and you can also see the advantages and disadvantages of each and every methods, like mechanical and physical methods have some advantages and disadvantages also. Similarly, the chemical and biological methods have also certain advantages and disadvantages. So amongst the above mentioned methods, these all are the method of the synthesis. So amongst the above mentioned methods, the green synthesis methods has emerged as a most beneficial method. What happened in green synthesis? So green synthesis utilize the different metals which have been applied in different fields such as medical, healthcare, etc. The biological metallic nanoparticles are synthesized by certain type of bacteria, certain type of fungi, certain type of algae to cure various disorders in medical fields for their antimicrobial activity, as a sensor for various biomolecules, for gene delivery, and for labeling of cells in medicine and plant. So this is the method for the synthesis of the nanoparticle. Now there are three methods as we already discussed: chemical synthesis, physical methods, and biological synthesis. And I already told that among all these three methods, a green synthesis, that is the biological synthesis methods, has emerged as a most beneficial methods. What happened in chemical synthesis? The chemical synthesis, there are hazardous chemicals are used, so ultimately it polluted the environment and detrimentally effect on the ecosystems. Similarly, in physical methods, there is a high energy consumption for the preparation of the nanoparticles. So compared to this method, chemical and physical method, biological synthesis methods are most beneficial methods. Now in this slide, I am focusing on biological synthesis of the nanoparticle. This method is widely used in any laboratory throughout the globe because it is very easy to perform and very cost effective. Okay, so to reduce the metal ion into nanoparticle, secondary metabolites secretion and intra and extra microbial enzymes play a very important role. You can see that in the plant extract, in the fungal extract, in the bacterial extract, there are certain metabolites are there are certain microbial enzymes are there, and these metabolites and enzyme play a very important role for the synthesis of nanoparticle, for the reduction of the metal ions. So under the metal ion stress, microorganisms secretes enzymes and biomolecules which reduce the effect of metal ions, and the toxicity of metal ions are then reduced by the detoxifications. So there are three steps which have been reported for the biosynthesis of nanoparticle by microorganisms, which have been shown in this figure. You can see the plants and microorganism at the top sides. You can see the fungi, bacteria, yeast, algae and actinomides as a microorganism, and plant tips, leaf, root system of the plant, agriculture waste, fruits and vegetables as a plant parts. What happen with the plants? So these plant parts have carbohydrates, proteins, vitamins, polysaccharides and organic acids, while at the microorganism sites it secrete certain proteins, enzyme and secondary metabolites. So these type of secondary metabolites and proteins play a very important role for the reduction of the metal ions. Okay, now we will see the metal ion and microbial interactions in the uh, flask. You can see in the figure there is a microorganism and plant extract. So what happened after the addition of the metal? So how the metal ions and microbes are interact with each other? So through electrostatic interaction, metallic ions attach to the negatively charged surface of the microbial cell wall and metallic and transported inside the cell through cationic membrane transport system. So the metal particles are going inside the cell of the microorganism. Now here the reduction is occurring. So metallic ions can be bioreduced either by functional group associated with biomolecules having a reduction capabilities or by microbial enzyme. The most important enzymes, most reported enzyme is NADH dependent nitrate reductase, which catalyze the reduction of silver ions. Here we are talking about the silver nanoparticle synthesis. So silver ions to the silver nanoparticles. So in these reactions, a mono or divalent oxidation state is converted into zero valent oxidation state. After reduction, zero valent state silver nanoparticle associate to form a various morphological shapes like oval, spheres, cubes, triangles, hexagons, etc. nanoparticles. So how the plants and microorganisms play a role to reduce the metal ion and produce the respective nanoparticles. And certain chemicals, certain metabolites, certain proteins, certain enzymes present in the extract of plants, fungi and algae play a very important role for the stabilization of the nanoparticle also. So this biological synthesis of nanoparticles plays a very important role for the successfully production or fabrication of the nanoparticles. After the synthesis of nanoparticles, we have to characterize that. So the here the second part of our talk is characterization of silver nanoparticle or characterization of the synthesized nanoparticles. There are certain you can see that here I quote two research papers. One is from Biotechnology Letters Journal, and which is honored by Springer Nature, and one is a paper which shows silver nanoparticle synthesis by the fungal extract, which having antimicrobial, antioxidant, larvicidal and anti-cancer potency. So such type of work, this type of work are published by my lab, means our researcher. My PhD students are working in this area since last 4 years, and we successfully synthesized silver nanoparticles and check their biopotential activity like antibacterial, antifungal, antioxidant, larvicidal, and anti-cancer potency. And this type of works has been published in very reputed journal. The below journal is a journal of applied microbiology, which having a impact factor 4. So such type of work is also right now it also carried out in my laboratory. So any professors, any teachers who is interested in the such type of work, they can contact me at HNGU Patan. So after the synthesis of such nanoparticles, we have to characterize that. This is the characterization. Now we will discuss about the characterization. So characterization have including many sophisticated instrument and processes. The first one, UV visible spectrometer. So to check either nanoparticles are developed or not, visual and colorimetric appearance of samples checked by UV visible spectrometer before and after formulation of nanoparticles at different time intervals. So we have to check before the reaction start and after the reaction start, but at a different time intervals. What happened before synthesis of Ag nanoparticle? Silver nitrate is colorless. Here the silver nitrates act as a raw material for the synthesis of silver nanoparticle. So before synthesis of Ag nanoparticle, silver nitrate is colorless, and once Ag nanoparticle synthesized, it develop yellowish brown color. You can see in the figure. Figure A is a colorless one and figure B is a yellowish brown in color. This is our research only, that's why I have put this figure. And after interacting with extract of the plant or bacteria or fungus or algae, which confirmed by UV visible absorption in the specific range of 400 to 475 nanometer. You can see in the graphical representation there is a one peak at the between 400 to 450. So it is a unique characteristics lambda max of silver nanoparticle. If you are working with the gold nanoparticle, the lambda max is different. If you are working with any copper, nickel nanoparticle, the lambda max is different. Here I am showing silver nanoparticle lambda max. It is lies between 400 to 450 nanometers. So this how the UV visible spectrometer is utilized for the characterization purpose. Then FTIR analysis also done for the further characterization. The FTIR spectroscopy is an investigational tool to determine or confirm functional group present in the moiety, which is characteristics of the dead compounds. We already seen that a nanoparticle has a outer layer on which certain functional groups are attached. So to know about the functional groups attached to this synthesized nanoparticle, FTIR spectroscopy has been done. So the major functional moieties or groups present on Ag nanoparticles were identified by scanning the samples in the range of 4000 to 400 centimeter inverse. Here in graphical representation you can see the peaks, downward peaks. These each and every peaks indicate some of the functional group. You can identify this peak to know the library, to compare with the library of the FTIR instrument. So this how the FTIR spectroscopy is also utilized for the characterization purpose of silver nanoparticle or any nanoparticle. Then another method for the characterization is scanning electron microscopy and transmission electron microscopy, that is SEM and TEM. We are routinely utilize a terminology as a SEM TEM. Okay, so this SEM and TEM mainly used to study the surface morphology of the synthesized silver nanoparticle. So what happened in SAM and TEM? So in SAM and TEM, plates were prepared by addition of silver nitrate to develop smear of solution on the slides. So there is a particular one slide, very tiny slide, and on which the layer of the silver nitrate, synthesized silver nitrate, has been prepared. Then conductivity was incorporated in system by making thin film of platinum which was coated on the slide. Once these slides are ready, they are scanned at 20 kilowatt, that is the higher voltage, that's why it is known as the electron microscopy. Okay, scan at 20 kilovolt accelerating voltage and high quality images were captured. Here you can see the four images A, B, C and D. The images A and B is from the SEM, scanning electron microscopy, and the figure represent as a C and D, it is the transmission electron microscopy. Here you can also measure the size of the nanoparticle by scaling background available in the software of SAM and TEM. So this is the third sophisticated instrument for the characterization purpose of the silver nanoparticle or any nanoparticle. The another one is the X-ray diffraction. So this X-ray diffraction is the modern technique mainly utilized to identify state of matter, either it is crystalline or amorphous. So we can know the structure of the nanoparticles, either it in crystalline form or either in its amorphous shape. So we can utilize the X-ray diffraction data to know the crystalline structure of the synthesized nanoparticles. So there are also certain sophisticated instrument techniques are known for the characterization of the silver nanoparticle or any other nanoparticle. I have seen only four methods which are primarily required for the characterization purpose. Another zeta potential is also there. Certain microscopy, AES, ICP are also can be done for the further characterization of the nanoparticles. But these four are very basic and very much needed for the characterization of the nanoparticles. So this how we already seen the types of nanoparticle, then methods for synthesis of nanoparticle, and how it is characterized. Now the most important part is biological applications. There are tremendous type of applications of the nanoparticles worldwide, but here we discuss few of them, means which are the most important, they are discussed over here. Okay, now the first biological activity, the most common, is the antimicrobial effect. Each and every nanoparticle have antimicrobial effect. So when nanoparticle reaches toward the cell, they release ions. If we are talking about the silver nanoparticle, you can see in this figure a silver is central part, and there are certain mechanism through which antimicrobial effect of the silver nanoparticle is exerted. So when silver nanoparticle reaches toward the cell, they release silver plus ions. This release ion, you can see this the formation of the free radical in the figure. So this released ion then interact with sulfur and phosphorus containing compound present in the cell wall. This lead to disarranged cell wall formation and small pits forms in the cell wall. These pits gives access to entry of ions and other foreign material to entry inside the cell. This increase intracellular osmotic pressure. As pressure built up in the cell, it begins to swell. Okay, the cell is begin to swell. Finally, all these events leads to bursting of cell wall and cell lysis take place. So this is the mechanism by which silver nanoparticle having a antimicrobial effect. So primarily morphology, that is the size and shape, along with reactivity of silver nanoparticle are responsible for bactericidal potential of silver nanoparticle. Size and surface are inversely proportional to each other, as we know. As size decreases, area increases. This leads to rapid rise in surface area to volume ratio. As we know, this is the unique property of the nanomaterials. So, bactericidal potential inhibit cell wall and free radicals Ag thiol groups, that is silver thiol groups of enzymes, preventing the biofilm formation. Here free radical is preventing the biofilm formation. It also intercalates between the bases of the DNA and prevent the expression of the particular proteins or particular products. It also attaching to the surface of the cell membrane and prevents the entry of some important molecules inside the cells, and it's also attached to the 13S subunit of the ribosome, prevent the protein formations. So these are the possible mechanism of silver nanoparticle for exerting is its antimicrobial effect. Okay, so this how the nanoparticles having a antimicrobial potentials. So this is the common one application of the silver nanoparticle. Now we are moving towards the another application, that is the antiviral potential. So each and every nanoparticles have these properties, but the mechanism is not well known. There are several studies based on the antibacterial property of the metal nanoparticle, yet the antiviral property of metal nanoparticles have limited reports. Some researcher, we can see the some researcher have reported on the antiviral property of different nanoparticles. Okay, so such type of metal nanoparticles have been proposed as an antiviral system exploiting the core material, and additionally the ligand shell studied the antiviral property of silver nanoparticle biosynthesized by the bacteria, fungi or algae. But here I am trying to show one important mechanism, one well-known mechanism as a antiviral potential. What happen when there is nothing, means no nanoparticles are there? In the first half you can see the virus is coming near the host cell and which make the entry inside the cell. So entry is via the genomic materials. So the viral RNA easily enter inside the host cell and it incorporate with the our host genetic material and activates its viral factor and cellular factor, ultimately leads to the viral replication and progeny virons. So these how virus are capturing the host cell. Now what happen when you utilize the metal nanoparticles or nanoparticles? Here in the second half of the figure you can see that the blue one is the metal nanoparticle. The first approach of this nanoparticle is they are interaction of the nanoparticles with virus glycoproteins. So once all the nanoparticles are bind with the glycoproteins, the virus is unable to bind to the host cell and it is prevented by the infections. Another mechanism is that once nanoparticles are penetrating inside the host cell, and if there is a virus inside the host cell, so this nanoparticle is interfere with viral genome, viral factor and cellular factor, and ultimately resulting in block the viral replication and no progeny of the virons are there. So this is the possible antiviral mechanism of the silver nanoparticle or nanoparticle. As I already told that antiviral properties of such type of nanoparticles have limited reports. So that's why this is the possible way of the nanoparticle mechanism as a antiviral potentials. Now moving forward to the anti-cancer activity. So today we hear about that out of 10 people, one people has affected or suffering from the any type of the cancers. So this nanotechnology have a tremendous application as a anti-cancer activity. There are many reports related to nanoparticles which are successfully cure the anti-cancer means cancer of any stage, but there is a limited mechanism. There is a very known less known of the mechanism of the anti-cancer activity. Now how this nanoparticle has go for the anti-cancer activity. So there are various mechanisms as mentioned in the research works through which the anti-cancer property of silver nanomaterials can be justified. So silver nanoparticles efficiently interact with cancer cells because cancer cells shows the enhanced permeation and retention effect, that is known as the PRE effect. So due to this effect, that results in the entry and accumulation of more and more silver nanoparticle insides. So ultimately what happen, more silver nanoparticles are penetrating in the cancerous cell, this leading to the death of cancer cells or hindering their uncontrolled division. Moreover, silver nanoparticle act by downregulating and upregulating the signaling physiological pathways, resulting in early apoptosis or checking the rapid pace of the division of tumor cells. Further, some research work suggested the activation of p53 caspase 3 by silver nanoparticle eventually lead to the apoptosis and regulate cell division through a series of events taking place in the cell. Here you can see inside the in the figure in the slide, the gold nanoparticle have a tremendous effect, a various mechanism by which the cancerous cells leading to the apoptosis or cell death. Okay, so these are the major activity at the biological science. The pharmaceutical industries widely use such type of nanoparticles in the research field and also include some particles in medicines also. So this type of medical research including antibacterial effect, antiviral effect and anti-cancer effect are widely utilized by the research groups of the pharmaceutical companies. Now we moving forward the another application like in environmental applications. So how the nanotechnology are utilized for the environmental industries. In the next slide you are seeing that there is a water and wastewater treatments are there. Now why it is important? So according to the World Health Organization, that is WHO, near about 844 million people, this is the 2020 data. Okay, so near about 844 million people still lack an adequate drinking water supply. The cost of the potable water is also increased due to rising energy cost, rapid population growth and climate changes or other environmental issues. So recent progress in nanobiotechnology may provide a new prospect for development of advanced water and wastewater technology processes based on the nanomaterials nature. We already have seen what is the nature of the nanomaterials. So they based on the nanomaterials nature, it may be categorized into two major categories. One is nano adsorbent and another is the nano membranes. Okay, so in this figure I have tried to show the adsorption technology. in which, like in our RO system, there are many filters are there. So these filter are fill up of certain membranes, certain chemicals like clay, biochar, chitosan, agro-industrial wastes, etc. Okay, so this type of materials has utilized for the filtration of the water and wastewater. So similarly, instead of this activated charcoal and biochar and clay, the modern technology, that is a nanotechnology, utilizing the nano absorbent material instead of that for the effective filtration of water and wastewater. So adsorption is a surface process which is generally used to remove organic or inorganic pollutants in water and wastewater treatment, wherein the pollutants are adsorbed on a solid surface. Now, nano adsorbents offer a large surface area due to the less size. It offers a large surface area, small size, and large number of active adsorption sites compared to the conventional absorbent. This unique property of nano absorbents make them ideal for wastewater treatment applications. These nano absorbents used for water treatment can be carbon based, polymeric, magnetic, or non-magnetic based and metal oxide based, including modified compounds such as oxide composites and zeolites. So this application of nanoparticle used as absorbent should be non-toxic with high absorption capacity and have sensitivity to low concentration of pollutants. It will be cost effective if pollutants could be removed from the surface of the nanoadsorbents easily, and sorbents could be recycled for the several times, and the process could be reversible to recover the absorbent. Factors controlling nanoadsorbents properties include size, surface chemistry, shape, crystal structure, chemical composition, and solubility. But this type of technology are still in the pipeline. Certain proof of concepts are there for the water and wastewater treatment, but much research about that is needed. The another one is utilized for the water and wastewater treatments are nanomembranes. Uh, as a biological person, we all know the lipid bimembrane. Okay, lipid bilayer. We know that the lipid bilayer. Similarly, the biomembrane is similar structure over there. It is a selective membrane. So nanomembranes are widely used in water and wastewater treatment and have replaced reverse osmosis due to the lower operational cost and reduced energy consumption, including higher flux rates. In addition, that is moreover, they also have high rejection for most multivalent ions and organic molecules with low molecular weight. So based on membrane structure, nanocomposite membranes can be categorized into two types. There are two types of nanomembranes. One is mixed matrix membrane and another is thin film nanocomposite membrane. So mostly nanomembrane are typically fabricated by interfacial polymerization on ultrafiltration substrate to form a thin film composite structure. Example is nanozeolite, nanosilver, nano titanium oxide, and carbon nanotubes, you can call it CNT, are generally used in these applications, and among them nanozeolites, which enhance the membrane permeability, are frequently used in thin film nanocomposite materials. Moreover, carbon nanotubes are used in thin film nanocomposite membranes for their antimicrobial properties. So, these nanomembranes and nanoabsorbents are widely used for the environmental purpose, especially for water and wastewater treatment. This nanotechnology or nanoparticles are also utilizing for the pollution removal. So nanotechnology plays a vital role in reduction of pollutants at the source by efficient use of raw materials, energy, water and other resources. This approach also includes using less toxic and renewable sources and processing materials by preventing release of contaminants produced during the manufacturing process. One example is very known that gold nanocatalyst can be used to oxidize carbon monoxide at lower temperature. Okay, so this type of nanostructures can be reusable and absorbs oil up to 20 times the weight of the material in preference to the water over a combination of superhydrophobicity and capillary actions. Example is TiO2, titanium oxide. Titanium oxides can be used in the breakdown of volatile organic compounds, nitrous oxides and other pollutants into less harmful products. So this type of nanomaterials or nanoparticles are also utilizing for the removal of the pollutants. The most important issue is the carbon dioxides. So typically most of the energy utilized in industry is generated from fossil fuels through combustion, thereby releasing higher level of carbon dioxide into the atmosphere, consequently causing the global climate change. A number of carbon dioxide capture technologies involving absorption, adsorption, cryogenics and membranes have been studied, but among them adsorption regeneration technology based on amine based adsorption is the most recognized and developed process. Example for the adsorption including activated carbon, zeolites, silica adsorptions and carbon nanotubes. Adsorbents such as functionalized single wall carbon nanotubes and multi-walled carbon nanotubes have received attention for CO2 capture due to their unique properties such as high thermal and chemical stability. So carbon nanostructures like carbon nanotubes are widely utilizing for the carbon capture from the atmosphere. Now nanoparticles has also a tremendous application in plant sciences, especially in agriculture side. A nano fertilizer, this one is the very much discussed part. So what happened with the nano fertilizer? So different methods such as plant breeding, fertilizer and plant protection products have been used for increasing the crop yield. The decline in agricultural productivity has been reported since the Green Revolution, which needs another revolution in the agriculture nanotechnology. The nanotechnology is a quickly emerging field with the possibility to advance forward the agriculture and food industries with new devices and tools which guarantee to increase the food production in sustainable manner and to protect crop from the various diseases. Now the nano fertilizers and meets these two aspects and play a very important role in agriculture by increasing the production rate and crop yield. Now what happened with the nano fertilizer, how it works? So the consistently growing human population is creating the pressure for the agriculture sector to fulfill their continuously increasing demands. Chemical fertilizers that are generally used for improving crop productivity have a major adverse environmental and ecological effect, as we know that it is a toxic. Nanotechnology, which utilize the small size of nanoparticle with unique properties such as higher absorption rate, utilization efficacy and minimum losses, may offer an exceptional opportunity to create a concentrated source of the plant supplements. Nanofertilizers are being synthesized by encapsulating the plant nutrients into the nanomaterials and delivering them in the form of nano size emulsion. The uptake and deep penetration of nanomaterials are facilitated by nanopores and stomatal opening in the plant leaves, leading to higher nutrient use efficiency. You can see in the figure there is a plasmodesmata like opening uh, nearby the cells, and this is the entry point of the nanoparticles. Plasmodesmata, which are nano-sized channel between the cell, facilitate higher transport and delivery of nanofertilizer. The increased efficiency of utilization cause significantly less nutrient losses of the nanofertilizer, which ultimately leads to higher productivity and nutritional quality of the various crops. In the figure, you can see that the movement through the plant, nanoparticles are trying to penetrate in the plant cell and exert their effect, because such type of nanoparticles are loaded with the plant supplement, plant nutrients. So different approaches, as we earlier discussed, such as top down and bottom up and biological methods, are generally used for the synthesis of nanoparticle as nanofertilizer. So nanofertilizers are generally of two types: macronutrient nanofertilizer and micronutrient nanofertilizers. Different macronutrients such as nitrogen, phosphorus, potassium, magnesium, sulfur and calcium encapsulated with nanoparticles reduce their overall requirements and deliver precise amount of nutrients to the crops. So these figures review the how the nanoparticle are playing a very important role for the food supplements to the plant, and it acts as a nanofertilizer. Now the another role of the nanotechnology and very important role of the nanotechnology is for the plant genetic engineering. So this is also a very important application of the nanoparticle. As we know that a cell wall represents a barrier to the delivery of exogenous biomolecules in the plant cells. It is the outermost layer. To overcome this barrier and achieve plant genetic transformation, different strategies based on agrobacterium transformation or biolistic method are worldwide used for the DNA delivery. We all are, as a biological person, well versed with the agrobacterium mediated gene transformation. So this type of transformation are worldwide used for DNA delivery in plant cells. Limitations to this approach, which approach agrobacterium and biolistic, rely on narrow host range and plant extensive damage, which often inhibit the plant development. So the most of the pioneering studies, means early study for the nanomaterial based plant genetic engineering, have been conducted in plant cells culture. The most important and well known example is a silicon carbide mediator transformation. So this silicon carbide mediated transformation has been reported as a successful approach to deliver a DNA in different calli. Which calli? Like tobacco, maize, rice, soybean and cotton calli. So here I am trying to show one most known example of the nanoparticle, especially magnetic nanoparticle, for the successfully delivery of the foreign DNA inside the host cell. A successful stable genetic transformation has been achieved in cotton plants via magnetic nanoparticles. Beta GUS reporter gene MNP complex, you can see in the figure, that is the MNP DNA complex. Okay, then pollen is there, and this complex were infiltrated into the cotton pollen grains by magnetic force without compromising the pollen viability. Through pollination with the magnetofacted pollen, cotton transgenic plants were successfully generated and exogenous DNA was successfully integrated into the genome, and it is also effectively expressed and stable inherited in the offspring obtained by the self line. So this how the nanoparticle, especially magnetic nanoparticle, play a very important role. Without changing the characteristics of the plant cell, it is successfully generated a transgenic cotton plants. So this is the tremendous experiment through which nanoparticles play a very major role in the plant genetic engineering. So these are the well-known example of the nanomaterial in the various life science sectors, including the biomedical, environmental science, as well as in the agricultural science. So I here conclude my talk. That is, so in this discussion, we have discussed detailed information about nanoparticles, such as their definition, types, synthesis, characterization, properties, and applications. Several studies have shown that nanotechnology play a very important role in commercial development. It is improving the everyday lives of human beings by increasing their performance and competence with daily objects. This technology has been used to provide a safe environment by improving air and water quality, and also provides renewable energy sources for the sustainable future. We need to find more breakthroughs and novel prospects for advances in nanotechnology to develop a world economy. Nanoparticles are used in several fields such as agriculture, electronics, food, medical diagnosis, and pharmaceutical industries. So, while while taking advantages of newly developed nanotechnology, the possible environmental, social, and ethical implications need to be considered. Such implications could be realized by scrutinizing several issues such as the safety design of nanomaterials, toxicity testing of manufactured nanoparticles with various organismal model, nanoparticles ecological impact, nanoparticles complete life cycle assessment. Currently, there is a critical lack of baseline data on the fate of many nanoparticles in the environment. Therefore, risk assessment and the impact of risk factor associated with their utilization in the environment and possible adverse effect on non-target organism and mammals, especially humans, are needed. Furthermore, finally, it is essential to have regulate frameworks that properly address and specify manage the potential risk of the nanotechnology. Uh, Pertinent educations on nanotechnology and nanoscience, along with new and innovative ideas and proper risk assessment strategies, will definitely help in fulfilling the potential of nanotechnology and nanomaterials to improve the quality of human life while elevating negative environmental impacts. Finally, as for the safety design, a common preventive strategies in re-engineering this nanoparticle is to conduct an in-depth evaluation of their potentially hazardous physicochemical properties in order to guide the safe by design processes. So with this sentence, I end up my talk here, and I am very much thankful to all of you to hear me very precisely. Thank you everyone.